So, oops, sorry, I think, okay. So at Blueprint, we have explored FEP quite a bit in different programs at different stages. And today I pulled two stories, one a little bit of early stage, one a little bit later. And one is it's not something that is very common. It's an application of protein FEP. It's somewhat anecdotal, frankly, but I thought it's an interesting story just to share. And the other one is your typical lead op ligand FEP type of work. But before I start, I want to thank both project teams, especially the MedChem leads Mike Burke and Kevin Wilson, and then Tom Deneen. With their cooperation, I think this impact of FEP was possible only. So I have a couple corporate slides, just bear with me. I just want to give you guys a flavor of what Blueprint is about. So at Blueprint, we have a robust and diverse portfolio focused on highly selective kinase, small molecule inhibitors. And we're interested in cancer targets and some rare diseases, as long as there's a clear genomic driver for the associated indication. And we also work on some immuno-oncology targets with our collaboration with Roche. Really the idea behind Blueprint is this combination of Blueprint library, which is a unique collection of small molecule kinase inhibitors that are exquisitely selective, and sort of combining that with a strong biology team that works as a target discovery engine, which constantly works on target ID and validation and gives us new insights about the biology of these kinases as disease drivers. The Blueprint library itself is a little bit more than the sum of its parts. So it provides tool compounds for the target discovery engine to interrogate the biology of these drug targets or new targets. And because the library is fully annotated kinome-wide, it also allows us to ask very fundamental questions about potency and selectivity, about kinases as a class of enzymes in general. But probably arguably most importantly, it jumpstarts medicinal chemistry programs. It provides, it provides high quality chemistry starting matter. So the library itself, these hinge binding scaffolds, they sometimes come from different literature papers, sometimes designed by our chemists, computational chemists, structural biologists. And these different vectors extending towards different selectivity pockets, we rather agnostically decorate them without actually having a specific kinase in mind, and then collect single point inhibition, percent inhibition data across kinome. So then eventually we have, okay, hopefully you guys can see my, so we have, a fully annotated and high quality collection of small molecules that essentially drug discovery, you can now start thinking about it almost an inverted problem where when a kinase becomes a target of interest, then the library becomes a lookup table. We can go back to the library and then we can do some library triage, and essentially we have already high quality chemical matter to start with, which helps us to advance very rapidly towards optimization stages where technologies like FEP have so far been particularly useful. So at Blueprint, we really started playing with FEP using the per compound licensing offered by Schrödinger. But then it quickly became obvious to us to be able to establish, deploy FEP, you kind of need to do some experimentation, some validation. So we bring in-house licenses, and we're relying on AWS for the GPU resources. And since then, we have successfully different levels of success, I guess, or different capacity, but we have applied FEP for over 5 projects internally. And typically we use ligand FEP to prioritize ideas for chemistry, for synthesis queue, or sometimes we just go through multiple rounds of designs and create some in silico SAR to increase confidence on particularly synthetically challenging targets, but could potentially provide high value. And occasionally we explore protein FEP as well. I think I would argue that in a high-paced drug discovery setting, properly validating, collecting data to properly validate protein FEP is a little bit more challenging than validating or benchmarking ligand FEP. Nevertheless, I think sometimes it comes as a handy tool just to use a little bit qualitatively, just to generate ideas. And I have one example for that in this program, and I'm not going to be able to share any structures today, but I think the concepts are there. You're not going to need to see the structures. In this program, the goal was to generate kinase 1 selective tool compounds, essentially. It was a very early program. But then we had to spare kinase 2, which had some overlapping, but also some distinct functions. And we determined, we had about 10-fold selective hits from our library, internal library. And when we obtained the crystal structures, matched pair of crystal structure of that 10-fold selective compound with kinase 1 and kinase 2, the structures looked very, very similar, almost identical. So there wasn't really much selectivity hypothesis from the structures themselves. So, I mean, I guess the question was, is it even possible to achieve selectivity from a rational design perspective, and how do we start generating these hypotheses? And sort of desperate times require desperate measures. So I wanted to explore a little bit protein FEP here, and there's probably millions of different ways to design a computational experiment like this. But what I chose to do was to really do an in silico alanine scan in the binding pocket. So I took both of my crystal structures, kinase 1 and kinase 2, co-crystallized with the same 10-fold selective compound. And I mutated all 25 of my binding site residues to alanine one by one, and I calculated the delta G. And not surprisingly, the profiles for two kinases are very similar. But then one residue, I mean, maybe a couple if you go down here, but then one residue stood out the most, residue 3 here. And like I said, so how do you know the cutoff here? You don't, like, what's the noise, what's significant? It's a little bit of experimentation, really, because we don't really know, right? I mean, in ligand FEP, we have our validation sets. We kind of know, our error margin from based on but here we're basically, it's a blindfolded experiment. But anyway, I decided to pick the residue that sort of that showed the most significantly different behavior in the two crystal structures. And sort of we decided, okay, well, perhaps we can just start our chemistry exploring that region a little bit. And we did, I mean, we built some virtual library, and, we were enriched for diversity and sort of your typical docking, clustering, whatever. And here the ligand FEP, we screened about 700 compounds with ligand FEP, but then the goal there was not really to pick the most potent compounds per se, or most selective compounds, but really to filter out the ones that are going to kill potency altogether. And the final set of 70 compounds for synthesis, that really was a combination of paying attention to the FEP scores, but also a little bit of medchem intuition. And eventually we got about 3 hits from that library, and they were roughly, equal selective, I guess. They provided some alternate vectors, some, they provided some path to follow. But I think the real value of this exercise really came from giving us an idea for targeted design, for focused design. So we at Blueprint, we deploy docking models through live design. So our medicinal chemists can tinker with their ideas. So actually Mike, the lead chemist in this project, he was able to, with this hypothesis, like he was able to design a compound. And he was able to improve the selectivity from 10 to 15-fold, which is, not too much, but we were particularly excited about the fact that he was able to do that in a 10-fold more potent context, because up to that point, if you increase potency, you lose selectivity. So that was the trend that we were seeing. So this was sort of a breakthrough. So I think the real impact of this exercise was really more qualitatively. It just gave us some ideas, some hypothesis to keep the chemistry going. And the other example that I have here is this program X, and this is on the other side of the spectrum. It's a late stage program. We were already in a chemical space where we had very potent and highly selective compounds, but we needed to fix some ADME issues while maintaining potency. And we were really exploring all of these pockets. And we were even playing a little bit with the core as well. And I think for the interest of time, I'm going to talk only about R1 and R4 pockets and what we've done there. The R1 pocket is back pocket. At that point, for the validation purposes, there was only 5 compounds available. And the reason for that is because we were pretty much locked in that chemical group there, because very small modifications were hurting potency very severely. So the chemistry team was a little bit reluctant to play with that. But then we kind of had to explore that vector as well for property purposes. So this set of 5 compounds, I mean, it's a nice, decent rank ordering, but it wasn't enough for us to feel confident for prospective use. So what we ended up deciding is sort of do a prospective validation, because at that moment, around that time, the project team decided to look into some back pockets, borrow some back pocket pieces from another program that had a similar hinge scaffold that provided a similar vector, and they didn't suffer from the kind of ADME issues that we were suffering. So we decided to evaluate some of those back pocket pieces. So I enumerated 52 back pocket pieces from that program into our scaffold here. And we also requested the intermediate without the back pocket for biochemical testing to increase the dynamic range. And then eventually we evaluated 53 virtual compounds here. But we wanted to de-risk compound selection. So we wanted to pick the top scoring compounds here, but also we wanted to make sure our chemists pick the intuition-based picks from the chemistry team are synthesized as well. So we didn't want to bias too much with FEP scores. So eventually 7 compounds were synthesized and tested. Well, one of them is the intermediate with no back pocket piece, but then the mean unsigned error was very reasonable. It was 0.9 kcal per mole. So this gave us some confidence, and then we sort of went on to the production stage, and we evaluated several different sets from different design principles, sometimes targeted design, sometimes bioisosteric replacements. I mentioned that we already had a very potent back pocket piece, so and we built libraries using commercially available reagents. So I mean, I guess overall we evaluated about 400 virtual compounds, and about 70 of them ended up in the synthesis queue. And I think of those 70, I tracked about 20 of them. And the mean unsigned error ended up being a little bit higher than the prospective validation. But I guess to be fair, these guys, the ones that, you know, four star, these guys were evaluated in one context, and the pieces looked attractive to the chemistry team, but then they chose to make those compounds combining with an alternate R4 group. So they're not really apples to apples comparison, but nevertheless, I think, I mean, they're shifted a little bit, but I think the relative affinity, I think it was reasonable enough. The other vector R4, we weren't that lucky from the get-go. So this was the initial validation set we had about 1.5 kcal per mole error, but this was a very critical vector for us, because we knew from our SAR that we can really pick up potency in this vector. So we really needed FEP to work well so that we can screen large libraries for this purpose. So I mean, we've done a lot of troubleshooting, and I'm not going to go through all of those, but eventually what we realized the problem was, after we requested an outlier, that FEP was underpredicting, but it was a very potent compound. So these two lysines, they're very critical to interact with a polar group in our series, lead series. And in the crystal structure, those lysines are interacting with our ligand, but also making some salt bridges with 2 aspartic acids. But in FEP, those salt bridges are overly rewarded, and the 2 lysines are essentially locked in this salt bridging network, and they're not really interacting with the ligand. So that's causing a little bit of underprediction. And just to test this very quickly, using the same force field, OPLS, I ran MD simulations, just mutating these aspartic acids to aspargines. And you can actually see, if you do weaken that electrostatic interaction, intramolecular electrostatic interactions, you do pick up your interaction with the ligand. So based on, after this quick experiment with MD, I reran my map, this time keeping the aspartic acids, but keeping them neutral, essentially. And then the predictions got much tighter, and that gave us a little bit more confident. And then we screened another 50-ish virtual compounds, I think, and eventually we we had about half a kcal per mole mean unsigned error. So I think overall, we were in this program, I think FEP really helped us to expand to a chemical space where it would have taken a little bit longer otherwise. And we have been able to do this across multiple vectors. It did require validation for each vector. And I mean, if you look at this table, you don't see R2 here, and that's because we actually never got FEP to work in the ribose pocket. For some reason, it just doesn't work. I mean, so sometimes it works, sometimes it doesn't. But we do know that the choice of reference compound and the crystal structures, like those are really impacting the final outcome, and not just from this program X, but also like our experience using FEP across different programs. I think this is just our learning. It's still a little bit in the production mode. It's still, even if you spend some quality time preparing your system and benchmarking, in the production mode, it still needs the data to be tracked, and the accuracy remains the similar level, and it still requires some hand holding, I think. So because of that, we haven't really invested much time in automation, but hopefully time will come for that as well. I'm very hopeful, actually. And finally, a couple points, I guess. Something that we've learned sort of as an organization, I think, for FEP to have a significant impact on a program, especially for tricky targets, that it doesn't work from the get-go, sort of a broader sense of commitment from the project team is needed to give FEP a decent shot to succeed. So being able to collect data to enable FEP that requires a different mindset, I think. So being able to request a retest of an outlier or a crystal structure of an FEP outlier, or being able to request a testing of a reaction intermediate. I think those are some of the decisions that, you know, the team needs to feel comfortable making today, because that takes away from their resources today, but it may or may not help down the road via enabling FEP. So that kind of a risk taking, depending on the team's comfort level, we have seen at Blueprint, it has worked for teams, but then FEP was not able to make that much impact on teams that were a little bit reluctant to make those decisions. And finally, I think this is the last part, is a little bit, I'm also curious, that's why I wanted to share with you guys. I'm curious about what everyone else is doing. So once FEP became a workforce for our portfolio programs, then we suddenly realized we're generating all this data, ever-growing data. So how do we share this data? How do we communicate with the chemists? And how do we communicate the confidence level in each set of predictions so that they can adjust what they pick from those scored compounds? And then how do we integrate data? How do we track the synthesis queue, and then what is made, and whether or not it was evaluated in the same exact context that it was synthesized? So we kind of relied on a combination of live design and NIME workflows to talk to different databases. But it requires some, you know, some attendance as well. So I'm wondering what everyone else is doing. I don't know if you guys would share to comment on that. But that's really all I have for today. And thank you guys for your attention.
Thank you very much. I'm sure that if there's any recommendations, they'll put in the Q&A. We're a little bit over the hour.
Sure.
You have 3 outstanding questions right now. I'm just gonna say one so that we can move ahead. Okay. This one comes from Emil Fichichi. Do you think improving FEP predictions with asparagine mutation might suggest alternative protection states of acidic residues?
Yeah, I mean, eventually, in the actual FEP, I did use the neutral aspartic acids, right? So, but then we don't have any experimental evidence that would suggest that is the case. So by sort of like physiologically, they needed to be, deprotonated. I don't think that's necessarily true, but also from the a comparison of a crystal structure and an FEP and a snapshot from an FEP simulation, like how those distances are like almost like 2.5 angstrom, whereas you're on the like order of 3 angstrom. It's, to me, it felt like a little bit more, it has to do with the force field than the actual protonation states in solution.
Okay, thanks. So let's thank the speakers of this session again for their interesting talks. And now I'll hand it over to Serena, who will moderate the second session of this meeting. Thanks again.
Hi, so we've got a break in the schedule now for half an hour.
Oh, there's a break, okay.
Yeah, so in whatever time zone you're in, I think if we start again on the hour, half past the time what it is now, in whatever time zone you might be in, and we'll start the next session that Serena will be moderating. Thanks.